Okay. Good morning, everybody. Thank you all for being here. This is our second Biotech Connector of 2026, and today's topic is engineering tumor organoid platforms for precision oncology. We're delighted to welcome you in person and all the folks online. I'm Maggie Scully. I direct the Partnership Development Office for the Frederick National Laboratory for Cancer Research. The Biotech Connector event is a collaborative effort between the Frederick County Chamber of Commerce and the Frederick National Laboratory. The event celebrates the Frederick County biotech ecosystem. It showcases local innovators and creates valuable networking opportunities for all of us. So I want to thank the Chamber for their continued effort. This has been a long-standing collaborative effort for many years. Now, given this life science audience, I wanted to take a quick pivot and also draw your attention to another local event happening on Hood College campus. It's called the Reproducibility in Science Symposium on June 16th to 18th of this year, so just a few weeks away. The event is intended to bring in scientific leaders to collaborate with the entire audience and collaborate in real time to build and reinforce standards that will help enhance scientific reproducibility. The registration is free, but it closes soon, so please act quickly. There's a QR code here, or you can register at hood.edu/standards. So now to our main event, I'd like to introduce our three distinguished speakers who will share insights on tumor organoid platforms. First, we have Dr. Nathan Coussens. He's the scientific leader of the In Vitro Evaluation and Molecular Pharmacology Laboratories here at the Frederick National Laboratory. His team is focused on the preclinical development of cancer therapeutics. Previously, he was a senior research scientist at the National Center for Advancing Translational Sciences, NCATS, where he developed biochemical and cell-based assays to identify and characterize small molecule modulators from high-throughput screens. Nathan is a member of the Frederick National Lab Scientific Standards Hub and the SLAS Technology Editorial Board, as well as an associate editor and former editor-in-chief of the NIH Assay Guidance Manual. Next, we'll have Dr. Paul Volto. He's the CEO and founder of Mimetis, which is an early pioneer in commercial organ-on-a-chip solutions. Paul invented the phase guide technology enabling the organoplate, the first commercial organ-on-a-chip platform in 2014. Paul and his team advanced in vitro modeling, tissue vascularization, high-throughput screening, and productization of cell biology. Prior to joining Mimetis, Paul worked at Leiden, Freiburg, and Bologna universities, Silicon Biosystems, and ScienceWorks. He holds a Ph.D. in microsystems engineering from Freiburg and is a fellow of the Netherlands Academy of Engineering. Paul will be joining us remotely, but we do have a representative from Mimetis here, Dr. Morgan Hellman, right there, wave your hand, who's the director of the US for Mimetis. So plenty of networking opportunities. Hopefully, Morgan, you can stay for the whole time. Terrific. Okay, and our third speaker is Dr. Matt Dallas. He has been part of the Thermo Fisher Scientific Cell Biology R&D team since 2012, developing products for cell culture, including media, supplements, recombinant proteins, and specialty plastics for a variety of cell-based models. His team recently launched OncoPro, tumoroid cell culture medium for patient-derived cancer models. Matt earned his Ph.D. in chemical and biomolecular engineering at Johns Hopkins University, focusing on glycoproteins and cancer development. So we have a terrific set of speakers. Please welcome our first speaker, Dr. Nathan Coussens.
Good morning, colleagues, and welcome. It's a great pleasure to be here with you today. I want to thank the organizers for giving me this opportunity to share with you, and I want to thank each of you for showing up here, and all of those of you online. Thank you very much for joining me. So you can see that today I'll be telling you a little bit about a combinatorial screen from a panel of patient-derived colorectal organoids. And I come from the Molecular Pharmacology Laboratory. And so within the Molecular Pharmacology Laboratory, we take an in vitro approach to assess potential therapeutics for cancer using a wide range of models for human solid tumors. And so to date, we've worked with 219 established and patient-derived cell lines that will grow maybe on a flat surface. as a single cell type or multiple cell type. So that would be a monolayer, or 3D spheroids of single or multiple cell types. And then we've actually worked to date with 258 organoid models that were generated by the NCI's patient-derived models repository. And so what we do is we'll assemble these different tumor models into panels based on their characteristics and the goals of a project. And we assess approved and investigational oncology agents alone and in combinations using high throughput screening. And we'll actually assess each agent at multiple concentrations so that we can achieve kind of a concentration response surface to interpret. It's a lot of data, so we have to rely on high throughput technologies in order to generate the data that we need for these projects. And we make the data available to cancer researchers throughout the world. We'll also perform some of our projects for NCI physicians who are interested in whether there might be new opportunities to combine therapeutics in their portfolio. And so how did we get into this? The history of our group actually goes back 40 years to the development by my colleagues of the NCI 60 production screen, which is a service for the research community. And it consists of 60 human tumor cell lines that were derived from 9 cancer types that are listed on this slide. And over the past 30 plus years, we've screened about 6,000 agents annually for investigators to assess their potential anticancer activity. And at the time that this was established in the early to mid 1980s, just at the beginning of high throughput screening, I think the goal was to develop therapeutics for these different cancer types listed. But it turns out that the model that we're using here of cells growing on a flat surface wasn't sufficient to predict patient responses. But something very unexpected happened. It turns out that when you assess an agent in such a large panel, many times the responses of the panel members to the agent can give you a hint of the mechanism of action by which the agent's targeting the cancer cell. And so you can't see it, but there are 60 cell lines listed on this heat map. And on the bottom, 1,003 approved and investigational agents. And what you can see from the heat map is that each of those agents induced a pattern of responses in the 60 cell lines. And the way that plays out is shown by this. It's a 3D so-called force graph. I had to compress it for the slide, but this little cluster of points corresponding to inhibitors of the epidermal growth factor receptor, or EGFR, they cluster together in three-dimensional space based on their pattern in the heat map over here. So this is a really important service that we do for the cancer research community. We continue to do that, but we're also very interested in vitro models that can better predict patient responses, which is why we're focused on organoid models. I'll tell you a little bit more about today. But you can see the amount of testing we perform, and we rely on a lot of high throughput technologies. We occupy 3/4 of a building on the Fort Detrick campus. And on the first floor, we have 2 very large screening systems, one of which I'm showing you pictures of here. I'm very proud of this system because it was designed and constructed in-house by my colleague, Eric Jones, specifically to do the work that we do. And both of our screening systems are outfitted with similar components. Here you can see a number of liquid handlers, detectors, robotic arms. They have similar capabilities and a capacity for about 1,000 microplates. And for those of you that don't do screening, this is a microplate, and they all have the same geometry that was established in the 1990s, but they have different number of discrete wells that are able to perform discrete tests. So this one can accomplish 384 tests in one plate. And actually, in this slide, you can see a microplate with 96 discrete test points. But the purpose of this slide is to show you one of the ways that we assess responses of tumor cells to an agent. And we actually leveraged the same chemistry that allows fireflies to glow. So this enzyme, firefly luciferase, requires ATP, which is the energy source for cells. And measuring the luminescent light enables a very sensitive means of detecting anywhere from less than 10 cells to about 50,000. And another advantage that we love about this approach for measurement is that it can be equally applied, the same assay chemistry, to cells grown on a flat surface or different types of 3D arrangements so that we can compare responses across very different tumor models. So I mentioned that we are doing a lot more work with organoids, and we have kind of a unique purpose in mind, right? People do many different things with organoids, but our purpose is really to see if they would serve well in a high throughput screening environment. And so we've received today, as of now, 258 organoid models from the NCI's patient-derived models repository. And I'm showing you a bar graph so that you can see that we've actually obtained samples from a wide range of cancer types. But really, we are characterizing these specifically to determine whether they would suit our needs for high throughput screening. And we have a relatively straightforward characterization process. The first question is, can we scale these up to the amounts we need to perform this work? And if we can, we cryopreserve a number of seed stocks that we can later draw. And then we will draw those and test the performance of these models in a high throughput screen with 166 FDA approved oncology agents at 5 concentrations. And then aside basically a simple grade of whether it's definitely well suitable, it's definitely not suitable, or it might be somewhere in between for a number of reasons. But over time, we've discovered a number of organoid models that actually are quite suitable for high throughput screening, and we've been moving them into panels. And our most mature panel consists of 32 colorectal organoid models that were derived from patients who were treatment naive or pre-treated from primary tumor sites or metastatic sites. You can see these were derived from patients ranging in age from their 30s to their 90s. And this age chart also shows a balance of gender throughout these ages. We've tried to obtain samples from as diverse patient population as we possibly can. And you can see from this slide that they feature a number of clinically relevant characteristics. So the idea here in selecting this panel is to essentially have something that could serve as a clinical trial on a dish and representative of a patient population that you might see in the clinic. And given all these different characteristics, you can imagine that there's a range of morphologies that we observe. So here's a picture of each of these models in the actual test conditions used to generate the data I'm going to show you in a moment. One of the concerns that we had was, due to our needs to be able to scale these up and test them in a relatively compressed time frame, are we biasing ourselves only towards fast-growing models? And we want to make sure that we're able to also assess more slower growing cancers and develop therapeutics for those as well. And so we measured the growth within the assay conditions of these 32 models. So it's all thrown there together in one graph for you. But all I wanted to convey from this is that you can see that there's a nice distribution of growth kinetics throughout the panel. So how do we use these things to test experimental and approved therapeutics? We deposit these organoids into the microwell plate using our liquid handlers that I showed you earlier, and then 24 hours later, apply controls or test agents and take a time zero viability measurement with that firefly luciferase assay. And then for the data I want to show you today, we actually had 2 additional viability endpoints, one at 48 hours and one at 168 hours, okay? But additionally, we had a completely orthogonal measurement of response. We're lucky enough to have a microscope with an array of objectives that allows us to collect millions of images from a single experiment. that we can use to generate live kinetic growth readouts of these models exposed to controls or test agents. And so these are very different approaches to measuring a response. How do they compare, right? So it turns out, in my opinion, and the data we've seen so far, that they actually compare really well. And what you can see in this slide with the yellow panels in total is 8 oncology agents. And the yellow panels show you that firefly luciferase viability readout. You can see it's very sensitive. We see reproducible results on a log scale. And in the green panels is the corresponding growth readout based on the imaging. And I want to emphasize that these data are from exactly the same samples and technical replicates, okay? And what you can see here, for example, with afatinib is a increasing responses to the increasing concentrations. At the highest concentration, nearly 3 log reduction in viability. Over here from the growth, you can see compared to vehicle shown in black, concentration dependent reduction in growth. And at the highest concentration, what looks like a regression. On the other hand, end of the spectrum, you can see in response to a range of oxaliplatin concentrations, we didn't observe any change in viability by our very sensitive readout. And similarly, within the error bars, not much of an impact on growth. So these are pretty concordant. You can also appreciate, I think, from these data that while we have very sensitive readout of viability based on an ATP measurement, it's less sensitive in measuring the growth. But we think it might be much more relevant to patient and animal responses, where responses to therapeutics is based on the growth of the tumor over time. So what we did with this panel is as kind of a proof of concept endeavor, we assessed 22 combinations of agents, including standard of care and selected agents, and in some cases had up to 3 agents in a well at the same time, but we would only vary concentrations of 2 of them, as shown in this slide right here. Now, this results in a lot of data. I told you 32 models. We have viability endpoints at time 0, 48 hours, 168 hours, plus we have live kinetic growth. A lot of data. Too much to unfortunately show you right now. But one of the things we can do to see this data set from a very high level is just look at maximum response. And so I'm going to show you that just for the standard of care right here. And it's a heat map where blue indicates a very strong response and this sort of light green indicates weak to no response. And you can see that these data are organized such that the treatments resulting in the most substantial responses are on your left, and those resulting in the weakest responses on the right, with the models that overall most sensitive to treatment on the top and those least sensitive on the bottom. So this is the standard of care agents and their combinations. And here is the same type of data in the same organization for selected agents. Now, it's pretty hard to interpret a whole lot from this very, very high level. So I also wanted to zoom in a little bit and show you more what data underlies these patterns. And so I just want to show you an example of some of the data from combining the MEK inhibitor cobimetinib with the RAS inhibitor RMC6236. And so this is data from 4 models, and you can see each harbors a genetic variant, oncogenic variant to the protein BRAF. It's V600E. And what you can see at the top, these are the viability graphs. So you can see a reduction in viability as we increase cobimetinib, the MEK inhibitor, and further reduction in viability when we add in that RAS inhibitor. And interestingly enough, for this combination, you can see that there's greater than additive or synergistic activity, which is most apparent through these grids in the center. Blue indicates synergy and brown indicates antagonism. And then at the bottom are the growth responses. Again, we can see concentration dependent reductions in growth that's consistent but different from the viability graphs on the top. Now, I want to reemphasize that these data shown at the top and the bottom result from exactly the same samples and wells. They weren't separate experiments, but just so that you can appreciate how that growth is assessed and how the experiments are performed, I wanted to further zoom in on this model 857933. And so this is actually data from the exact microplate that generated the data we just saw. And what you can see in the lower left corner is vehicle treatment. And along the bottom, increasing concentrations of the RAS inhibitor. Along the side, increasing concentrations of the MEK inhibitor. And this is our combination matrix with different concentrations of the two agents and the highest concentration of both agents in the upper right. So you could see different extensive growth of these organoids based on the treatments that they received. And so we can quantify the area and show how that changes over time, which results in the data you saw in the previous slide. So I've run out of time, but in summary, I think that we've developed a really nice collection of colorectal organoid models with the hopes of allowing that to serve as kind of like a clinical trial in a dish for a number of projects. And as a proof of concept, we performed a screen with 22 combinations. And you could see that there's good concordance between our viability measurement and our growth measurement. And I showed you some of the very interesting data that resulted. These organoid models were derived from patients that also donated samples that enabled the generation of xenografts. So we're actually conducting tests of the same combinations in the corresponding patient's xenograft model so that we can assess translation between in vitro and in vivo. This is a huge amount of work that I just showed you. So these are the folks that did that work. And so I want to acknowledge all of their efforts, and I want to thank you for your attention. I'm happy to take any questions you have.
So you've got two measurements that are essentially telling you the same thing.
Yeah.
How much longer are you going to use the luciferase readout? Are you thinking about switching entirely to imaging-based platforms?
It would save a lot of money if we did. And in the end, our goal is for translation. And I mentioned to you that it actually might translate better for the growth. One of the challenges that I didn't have time to tell you about is that growth metric works really well for some models. And I gave you some good examples of that. For some models, it's pretty compressed and the data aren't as easy to interpret. And so if we only went down the growth route, it would exclude some models that have very important characteristics that we'd like to assess. The other thing is we don't necessarily have the data yet to say that the imaging results in better translational data. So those experiments are ongoing. We would like to know the answer to that. But at this moment, I feel like they're very complementary. And one's extremely sensitive with uncertain translational relevance, or at least uncertain how you translate that. The other one is more straightforward, but of lower sensitivity. That's how I think about it right now. But we have, I didn't show you this either, but we put all the data together, 32 models, 22 drug combinations. And actually the correlation between the imaging data and the viability data was excellent across the board.
Great.
Yeah.
Great stuff. I had
Thank
a
you.
quick question. Have you, or does somebody else, or you guys would be the ones to do it, but the comparison of this combination screen in organoids to some of the just, two-dimensional flat plastic colorectal cell lines out of the NCI-60, see how different it looks?
Yeah, it does look different and similar, and it seems to be model and drug dependent. You brought up the NCI-60 as an example. I mentioned that these organoids have corresponding cell lines, right, that were derived from the same patient tissue. We've also looked at organoid responses compared to the spheroid responses. And Overall, I think that there's quite a lot of agreement between those two 3D models. We haven't looked at it grown as monolayers, but that's an important question. But we did see some discrepancies that seemed model and drug dependent, and we haven't had a chance to investigate that deeper yet. But it's a really important point: is what are the strengths and limitations of these different models that I described to you? Yeah. Hi. Yeah.
Is there any notion to sort of look at the opposite, sort of develop a screen for like safety? So in other words, if your viability goes down with a certain drug for healthy organoids or cells, that might be an indicator, early indicator that this would fail in the clinic.
Definitely. That's a really important question. I mentioned that we do multi-cell type spheroid experiments where we have malignant cells and we have normal cells. And with those, we'll typically run controls where we just have the normal cells. And that provides us with some level of therapeutic index. For the organoid models I've described so far, we don't have healthy organoids to serve as controls. But we are planning on doing a lot more work with normal organoids and developing some that could serve as healthy tissue controls. Thanks for the question. All right, folks, thank you.
Okay, Paul, you can share your slides now.
Sure. Is this visible?
Yes. Can you hear us?
Yeah, absolutely.
Awesome. Morning.
Good morning.
Oh,
Yeah, is it
we can
visible?
see your slides though. We just saw
cannot
them
see-
and then they disappeared.
Okay, so maybe I need to do the full screen first
Are you
and then
sharing?
share. Just a second.
Mhm.
Yes, is it visible?
Yes, that's great. Okay, go ahead.
Great. Well, thanks a lot for inviting me, and I apologize for not being on site, but I have my colleague Morgan representing me for any discussions afterwards. I'm going to switch gears a little bit from organoids to organ on a chip, and actually also the combination of those, and with a focus, I would say, on capturing the complexity of human physiology in health and disease. And I just show a slide here which really summarizes, our view as to where the whole space is going. And that slide is this one. You see here two pictures. And one of these pictures is a model, and the other picture is the real thing. In this case, a liver slide. And of course, you can see that on the right-hand side, it's a liver slice from a human. And on the left-hand side, it is the how a liver is actually looks in some of our models, right? Where you have actually the vasculature and the hepatocytes and so on, and the stellate cells. And you can actually appreciate that we're getting awfully close to the real thing, even though we're doing this in the laboratory. Another nice thing is, of course, that these vasculature is then perfusible, right? So you can actually add immune cells and even circulating tumor cells, if you will. So for us, this is really the benchmark as to where we think the space is going, and how we need to look at cell culture going forward. Now, as a company, so I'm representing Memetas. Our our vision has been built on an organ-on-a-chip platform, which has a number of characteristics that I've summarized here in this slide. So as you've seen already from the previous slide, perfused vasculature is really a crucial element of cell culture going forward in our view, and that's something that is largely lacking in how we've been doing cell culture today. The other element that we find important is the comprehensiveness, right? The interplay between cell types, such as the epithelia, the connective tissue, the immune cells, because we believe that in healthy systems, homeostasis can be achieved in vitro by the interplay between those cell types. And if you disrupt that homeostasis, you get insights into disease that you wouldn't get in for instance, monocultures. Now, if you go to the other part of the slide, you see here two technical elements, no artificial membranes and unmatched throughput. So no artificial membranes means that we layer cells, gels and perfusion flow without making use of artificial membranes. We use surface tension techniques. And that has a benefit that cells can actually migrate, they can interact, they can self-organize, and they can remodel the microenvironment. So we call it a perfect balance between engineering control, because we do provide some control over what is going to be seeded where, and biological self-organization. Another advantage of our approach is that the throughput is really sheer unlimitless for cell culture standards, right? So we've been doing screens of up to 10,000 chips already, and we actually have never really been tested for how far we can take this. The one that I didn't discuss, the one in the middle, is the shipment across continents. So we appreciate that cell culture is difficult, inherently difficult, and the more complex these models become, the more we get confronted with the limitations of very often the quality of cell culture that might happen. And so we were thinking of, can we actually do that for the client, prepare the biology, and then ship it out to the laboratory of that client that only needs to take off the blister and start adding their compounds. And that's what we do as well. So we ship a number of our organ models live to clients. As I already said, so we build our platform on the organoplate, which is a microfluidic platform. It has 32 to 96 chips. It fits in all the robots, microscopes, and so on. And we induce flow by passive leveling, basically by gravity, right? So we put it under an angle. One reservoir becomes higher than the other, and then it levels out, and then you change the angle, and it levels out again. So it's a very simple way of applying perfusion flow. But if you put, for instance, 16 of these plates on one of these organoflow rockers, you perfuse thousands of chips on one rocker with exactly the same flow conditions, which is of course hugely important for the robustness and the reproducibility and the comparability between experiments. The organ models are based on kidney, brain, gut, lung, liver, vasculature, and of course also tumor models, which might also be around other organs. I'll give a few examples today. Some of them have been productized as organ already, others have not. The assays that we run are basically everything that's out there in the laboratory, from high content imaging, gene expression analysis, Luminix panels, name it. And sometimes we need to develop our own instrumentation because it just doesn't exist, like for instance the organoteer machine, which allows us to measure electrical impedance of tissues, so typically barrier tissues, and measure the barrier function of for instance gut epithelia, and then measure the response to toxicants or inflammatory stimuli. The data and screening is really, limitless, as I already alluded to. We use robots where needed. We use high content imaging and AI to interpret the data, and we support our clients with understanding disease better, discovery of compounds, ranking compounds, and also ADME-tox studies. Now, before I go into the tumor models, I want to start with a healthy model of the colon organoids. There was already a question from the previous speaker, right? So can you also look at adverse effects of tumors and of anti-cancer drugs? And of course, we know that anti-cancer drugs very often have effects on the colon and typically inducing diarrhea. And so we have developed a colon organoid model, which is based on adult stem cell organoids. And the previous speaker already alluded to that, right? But adult stem cell organoids, what happens is you take a piece of the primary tissue of the patient. In this case, you isolate the crypts of the gut in which are the stem cells, and then you stimulate the stem cells with certain factors such as wind, and you inhibit BMP to prevent differentiation, so that actually these stem cells start expanding, and you can actually start banking them, and you can also switch medium towards differentiation, and then they differentiate. So they keep all the characteristics of human epithelium. They also keep all the characteristics of the donor, but you can expand them and you can bank them and you can use them over and over in the same context. Now we take these balls that forms, which are called organoids, and we make them into a tubule. So we put them against an extracellular matrix gel. In this case, it's collagen gel, and then they form a tubular structure. There's flow going through that lumen of the tuber, which is the apical side, and we have a second perfusion flow, which allows us to sample on the basal side. And this is how it looks like in confocal microscopy, right? So this is a nice three-dimensional tubular structure. We use this to test for gastrointestinal toxic mechanisms. So we used 28 clinically relevant compounds that half of them have induced toxicity in the gut and the other half do not. We add that to these colon organoid tubes, apical basolateral exposure for 5 days with 1 redosing, and then we measure cytotoxicity based on cell titer glow. So it's ATP that we measure here, and we measure TER, which is an electrical resistance value. So basically the loss of barrier function, as we can measure with our Organoid TER machine. And what we can then see then here, if we dose this for 28 compounds, 14 of them have gastrointestinal toxicity effects. We actually pick up the majority of those. Some of them we do not pick up. We have 11 of them we do pick up, which gives us a 79% sensitivity. And of those that are not toxic, we pick up 13 out of 14. So that's a 93% specificity. So it's a really nice model that you can use for looking into these GI toxic events. And the nice thing is it's just shipped to your bench, so you don't even need to set up the colon organoid culture yourself. It's just, you take off the blister and you start measuring in a very simple way. Switching gears more towards oncology, so I just wanted to highlight here another model which actually was done by not by us but by one of our clients, Takeda, and was published, which also uses the gastric epithelium in a healthy shape, but now added also endothelial cells and some connective tissue in the gel, and compared that to the same model, but now not with healthy epithelium but with cancer cells, and they were actually looking for binders that actually give them the largest window, right? So you see here that at a higher concentration for binder Y, you actually see a very high level of LDH release corresponding to cell death, and a much lower level in the healthy tumor. So that is, I think, a nice example of how you can actually not only look at efficacy but also at toxicity and determine your window on it. Shifting yet another gear, we go to the more complex models where we actually include more of the microenvironment, and that starts with vascularization. We just recently launched our Organo Access platform with the Uniflow chips, which actually allow us to build vasculature that is actually long-term stable. And the reason why it's long-term stable is because as I explained to you, we induce flow by passive leveling, right? On a rocker, we change the angle, and of course you get nice flow and very, very robust reproducible conditions, but the flow is going into two directions. And that's actually not good for microvasculature. So we developed a new technology, our Uniflow technology, which instead of, which still makes use of the passive leveling with the same organoflow rocker, but instead of having the flow going up and down, it sort of recirculates dependent on the angle of inclination. So it goes through the vasculature under one angle, and if you then change the angle, it takes a second flow pass. So it always goes in the same direction. It's very good for the vasculature. So you see here a nice picture in green of perfused vasculature with immune cells migrating to it. You see here how we use that for grafting tumor tissue into the vascular bed. And here on the right-hand side, how we can actually add epithelium also on top of the vascular bed. And I'll allude to that a little bit later. So this is how these vascular beds look. They're actually mixed with, in this case, fibroblasts, long fibroblasts, and they form this vasculature themselves. In the side channels, we have a blood vessel and they connect, so you can actually perfuse them. And for us, it's important that we have this access to these lumens. So how long can we perfuse them? And in this example, we actually managed to push that to 56 days. So you have a healthy vascular bed for 56 days, where you can do things with, like, for instance, inducing inflammation, right? So adding, in this example, PBMCs. And I have some beautiful pictures here of how that looks like if you then add, for instance, CXCL12, so that they are actually extravasating from the vascular network into the tissue space. You can also add epithelium on top. So this is an example of primary lung epithelium that is on top, that polarizes beautifully, that we can airlift. And of course, in the context of oncology, it's interesting to look at the combination of these things. So in this case, it's also it's a tumor line from lung epithelium on top. And then we have T cells added to the vasculature, and then we stimulate that with CXCL12, and then we look actually at the T cell counts in these tumor models. You can also mix your tumor cells in the vasculature, and we did that actually for 16 donors in the context of an ovarian cancer project for Astellas in Japan. So we actually did pathology on these tumors. We isolated all the cells, so the CAFs, the CAMs, so the associated fibroblasts and the macrophages, the tumor, and we added into the vascular bed. And as you can see, right, the model recapitulates nicely actually the donor phenotype in terms of the amount of epithelium that you can find back and the amount of stroma that's in that tumor. This is a zoom in where you can actually see that you really start creating this microenvironment, which is very, very complex, where you can actually see that the macrophages are embedded in the tumor, that the vasculature is actually very close to that, and you can start looking at therapy responses in this complex environment, which does require enhanced image analysis, because of course you need to differentiate between the healthy part of your tissue and your tumor tissue. So we use Encarta software for that, and AI machine learning algorithms to differentiate between those tissues, and you get very nice responses to standard of care in this case. In the interest of time, I had 15 minutes, so I think I stayed within that time. I want to thank the team in Leiden, the team here actually around the corner at Frederick in Gaithersburg, and there's of course also the team in Japan. And with that, I'd like to thank you for your attention.
Any questions?
Very impressive work. I'm just curious, the more complexity you layer into your models, presumably the more difficult it is then to reproduce those models from day to day, or from experiment to experiment. Because the more complexity you get, closer and closer to actual human beings, which we know are not predictable. So how do you ensure that your more complex models with the vasculature and the epithelial and the immune cells, that you can kind of create something that is translatable from day to day, from month to month?
Yeah, really good question. Thanks for that. So I think there we need to distinguish between the biology and the technology, of course. If you look at the technology, what we typically find is that the complexity increases the robustness of the models, and very often also increases the reproducibility of the models. That is a bit counterintuitive, but my take on that is this micro homeostasis that I alluded to. So you actually get crazy small error bars if you look at reproducibility between experiments from a technical perspective. Now, there's of course also biological perspective, and there it comes down to things like, how many cells can you get out of a tumor? Like if we take the example of a primary tumor, how many can you bank? And if that donor runs out, how can you, how does the second donor going to respond? And that can actually be very, very different. So you capture donor response very specifically, you capture, but the fact that you reconstitute it allows you to put in a lot of quality steps, for instance, normalizing the amount of epithelia, normalizing versus the amount of vasculature versus the amount of stroma. So there are lots of tricks that you can do, but of course, and you know that like nobody else, I guess, right? The availability of sample and the logistics leading to that sample is always a challenge, and that's of course where the organoids are very, very, very good. So the epithelial organoids, because you can bank them and you can standardize them, but you can also still combine that with vasculature and connective tissue, of course. So I think it is part of the progress that we make this better and better and get our logistics more and more under control.
So maybe along those lines as well on complexity, because you were saying about like introducing the different cell types. So as we get more complex in cultures, and as you were saying, it's quite interesting and it's quite useful. But one of the limitations that I have found is media, cell media. I don't know if you have encountered any of those issues with your devices. We have seen that sometimes the cell media may provide like enough nutrients for a specific cell type, but it might be deficient for a different cell type, especially as we introduce these stromal cells or these other macrophages or other cell types. Have you seen that in your devices? So you think like maybe by introducing that flow, are the other cell types secreting enough the growth factors to maintain their homeostasis that you were like talking about? I don't know if you can maybe say something about that.
Yeah, no, absolutely great point that you are making. So I totally agree that growth media optimization is a crucial factor in this whole thinking. And to be honest, it's also something that needs to be rethought, I guess, right? Because the classical media that we have always been using have been optimized in a certain context, like fast growth of tumor cells, for instance, or stimulating just the epithelia in the case of the stem cell organoids. And so if you start pushing boundaries of building these more complex models, you need to optimize your media for those specific conditions. And the way we do that more and more at Memetas is we use this design of experiment strategy where we actually list the most important parameters that we believe are of importance to these media and optimize and figure out what are the optimal conditions for all the different cell types. In general, I would say that the media should become poorer rather than richer in growth factors when you compare it, for instance, to these very, very rich organoid media. And that is, of course, also intuitively understandable because these various cell types, they form a sort of a feeder layer, which is what in the essence was what the organoid medium is replacing, right? So the fact that it becomes poorer is not a strange thing. But I totally agree with you that optimization of growth media is of crucial importance for this type of cell cultures.
Other questions?
Thank you, Paul, so much.
Thank you.
Okay.
All right. Good morning, everybody. Thanks for being here and thanks for the opportunity to speak. My name is Matt Dallas. I'm part of the R&D team supporting cell biology at Thermo Fisher Scientific. I've been with the company for 14 years and lived in the area for just as long. It's really a thrill to be here. I want to thank the Frederick National Lab and Biotech Connector for the invite. Gotten a lot of kind of preview to some of the things I'm going to talk about, so let's just jump right in. And before we do that, I do want to get to some vocabulary. There's a lot of terms out there for cancer organoids, patient-derived models. We're going to use tumoroid throughout this talk. They all mean the same thing. The critical thing here is this is a means to an end to preserving critical phenotypes in cell culture-based models. Those phenotypes could be mutational profiles, they could be gene expression patterns, they could be expression of particular biomarkers, but that is the critical piece, is we want to preserve those phenotypes to better study cancer in a dish. All right, so the title here is a really an important one, and I think it would have been the same if I was here 10 years ago or 5 years ago. These tumor organoids, cancer organoids, or organoids in general continue to emerge as next generation cancer models. And I think, Nathan, you did a nice job of kind of setting the table here. The materials we've used, the immortalized lines grown in 2D, have been the workhorse of cancer research for a long time. But as we think about and examine those models more, it's becoming apparent that we need better stories. So if we look at mutational profiles, these lines often are hypermutated. They've spent decades in culture, and a lot of time can lead to accumulation of both relevant and irrelevant mutations. If we look at gene expression patterns, the story is the same. Tumor cell lines don't look like cells from patients. And this may help to explain why we collectively, as a scientific community, haven't done a great job of developing anti-cancer therapies. We do a really good job of killing cells in a dish, and we can do a pretty good job of killing cells in mice. But when it comes to human cancers, the story is a bit more complicated. Now, the question is, why for the last 5 or 10 years have these models remained emerging and not taken over some of these other long-serving models? We think there are 3 major bottlenecks. One, the media to maintain these cells in culture is very complicated. There are few commercial sources available. And so what that means is you got to go to 6 or 10 different vendors, buy individual components, and make them in your labs. It's really taxing. And actually, we have heard large biopharma, in some cases, are using full-time employees strictly to make media for these systems. It's not really a good use of those scientific skill sets. So that's bottleneck 1. 2 is the culture of them is more complicated. Growing cells in 2D in a monolayer is easy, admittedly. And these organoids generally need to be embedded within basement membrane extract. We have a product called Geltrex. You're probably familiar with Matrigel. They need to be embedded within that to function appropriately. And so it's just harder to do. It's labor intensive. And third, and this is a critical one as well, is restrictions in IP. So the inventors of the original organoid technology have done a really nice job of protecting those inventions with granted patents here in the US and all over the world. Now, if you're an academic, this is not a huge bottleneck, but if you are not in the academic space, accessing these technologies can be either really expensive or impossible, depending on the licenses that may be available to your organization. So these are, I think, 3 critical hurdles that need to be cleared before these move from emerging technologies to core to the way we study cancer. And it's with these 3 the hurdles that we have recently developed and launched OncoPro tumoroid culture medium. So this came to market in the summer of 2023. We're approaching the 3rd year anniversary. The rationale here is, 1, we wanted to optimize this for cancer. We've not really hit on this yet, but the original organoid media products weren't really products. The media designs were built with adult stem cells in mind and were later adapted to cancer. And actually, that poses some problems, especially when you're trying to derive new models, because if there are contaminating, or there always are going to be healthy epithelia in any cancer sample, we actually hear lots of stories about those healthy cells kind of paradoxically outgrowing the cancer cells in traditional organoid media. We don't see that in OncoPro because it was built for cancer. 2, I think we've done a lot of work to streamline the media prep process. What you see in that image is essentially what you need to grow colorectal tumoroids. And if you want to do other indications, there's actually some small text that's a bit of an eye chart for you, but there are tissue-specific factors that we would recommend to make this system compatible with lung, with breast, with pancreas, and other cancer types. So it is a core, kind of a blank slate that you can decorate depending on your areas of interest. And we've also done some work to streamline the culture method, and I'll talk about that in a little bit on a slide. Let me start with the easiest hurdle is on the media prep side. So on the right side of this image, you see everything that you need to make traditional organoid medium. Maybe understanding why you need full-time employees. What is not really pictured here is the need oftentimes for conditioned media. So you got to set up a whole separate cell culture system to produce some of the factors that enable traditional organoid approaches. On the left is what you need for OncoPro. So this becomes not a full-time job, but something the scientists can do in their standard course of work. Making this media is very much like making other kind of standard formulations that you and your scientists are comfortable with. On the culture approach side, I already mentioned these cells generally, historically, have needed to be embedded, encased within these matrix layers. That is perfectly compatible with OncoPro. So if you want to do embedded culture, OncoPro is entirely supportive of that. The problem with that, though, is the labor intensivity. So it is very difficult to use these culture methods in anything like an automated system, which means that you're doing a lot of manual pipetting. And if you want to make a large bank, you want to do a large screen or create cryopreserves, you've got to put down like literally thousands of gel domes manually, largely manually. So it's a ton of time in the hood. OncoPro also supports a suspension culture approach. So it's much like what Nathan talked about when you plate cells in assays. A lot of people are doing this in suspension. You can just do it without the encasement of the matrix. We've developed a method that allows you to expand the cells in that form as well. And so scaling up becomes much more simple. You're spending less time in the hood. I think critically, you're using less material, not just basement membrane extract, but less plastic, and allowing the scientists to focus on the things that are truly most meaningful, spending their time answering key questions, designing experiments, instead of slaving away in the hood. All right, so I'm not going to talk much about the patient-derived, actually not at all about the patient-derived models repository, but Nathan, you kind of set the table there. It's a really valuable resource for cells. These cells are hard to come by, and the IP restrictions I talked about earlier are actually a bottleneck for getting cells in your lab. The PDMR is a really nice way to do that. We've done a lot of work with OncoPro in a large swath of cells from the PDMR and shown really good compatibility. If anybody wants to talk about that, I'm going to show my email later. Feel free to drop me a line or grab me here before I leave today. So I think there's some overlap between what the NCI has done and what OncoPro enables. Beyond that, though, if you want to have models of your own, derivation is the key. And in order to derive new models, you have to have access to fresh tissue, which unless you're at a research hospital, can be another limitation. We have dealt with this through commercial sources, but I do want to acknowledge the derivation process is challenging. Rule of thumb is for every 2 fresh tissues you get, about 1 can result in a tumoroid. You might hear different success rates reported in the literature. What we deem as success is a pretty high bar. So these are not just cells that will aggregate and we can culture for some period of time. These need to be long-lived cultures. So for us, they've got to grow appreciably. We got to split them with some regularity. If we're not passaging these once every 2 weeks, they're just growing too slowly for us to make use of. And then critically, they've got to be freeze-thaw competent. We've got to be able to come back again and again. Just heard the comment about, hey, when fresh tissue runs out, what do we do? If you have cryo stocks, that problem is solved. On the right side of the image, you see some examples of what these look like, both in suspension. You can see these top images are all in suspension. These ones in the bottom right corner are under embedded conditions, but you have varied morphologies. That's really driven by the biology of the samples from which they were derived. So in some cases, you get large aggregates. In some, you get smaller grape-like clusters. Up here, you see a cystic morphology, so essentially hollowed out balloons. These are consistent over long-term passaging. Again, they're donor specific, and sometimes we will see actually heterogeneous morphologies within a given sample, and think this is some good evidence that we are capturing some of the heterogeneity that is inherent to these samples. All right, so once we can grow them, we can bank them, great. But if they don't look like the patient samples, they're really not of value to us. So I'm going to do a little bit of work here to describe how we are characterizing these and what we see in the system. So this slide is summarizing the mutational profiles. We're using a comprehensive cancer panel. This is looking at about 200 genes, thousands of loci across those genes. The long and short of it, each of these paired columns, on the left side is the tumor sample. On the right is the model that has resulted, the culture model. And you see really good concordance of these driver mutations. Now, critically, this is not just about can we call the mutation, is it present or absent? We're actually looking at allelic frequency. I'll show a slide and a couple. I'll show what that data looks like. But we see really, really good concordance. And I think critically, it's not just that in the short-term culture there's concordance, but this persists over very long-term culture. And I can show you that in a couple of slides. The other thing to note here is just the diversity of driver mutations. We are not specifically selecting for KRAS or P53 or any other mutational profiles. This is really, again, driven by the biology of the cells from which these models were derived. On the gene expression side, story is very similar. So here we're using a full transcriptomic assay. This has really become a workhorse readout for us. We are seeing something like 80 to 90% concordance in RNA-seq profiles, full transcriptome with the starting material. Now, that may paper over, it sounds fairly high, that may paper over some fundamental differences between the tumor tissue and these tumoroids that I do want to highlight. And the best way to do that is here with this volcano plot towards the middle right. There are something like 200 to 300 genes that are consistently differentially regulated in tumor samples versus the tumoroids. And when we look at that, you can see the KEGG pathway analysis down here below, it's fairly obvious what's happening. OncoPro is built for culture of these epithelial cells in the tumor samples. We do not preserve immune cells, fibroblasts, other stromal cells. And what we see is a dropout of genes expression associated with those cell types. So this is not a surprising result and explains some of the difference between why we're not seeing closer to 100% concordance. Now, I mentioned long-term culture, and maybe I'll start on the right side here. So this is a principal component analysis, again, of the full transcriptome. So it's taking 20,000 or so genes and compressing them into a small enough data set that we can visualize. And the take home here is that the proximity of each of these data points within a given color is quite close. So even over 50 passage studies, we are seeing minimal amounts of changes in the RNA-seq profile. So once we see that, say, 80% change from the tumor sample to the tumoroid, it is highly stable over very long culture periods. So you can easily distinguish between these three colorectal adenocarcinomas. Similar story with the non-small cell lung cancer models. Now, I do want to emphasize the point here is not to say you can take these out to 50 passages. These act and behave, and we essentially treat them like cell lines. And with that in mind, the guidance we would have is the same as I would for any cell line, is grow them for 20 or so passages and go back to cryostocks and start fresh. Something that has been really stark as we've gone through the development process and supporting this product post-launch is it's not just about the culture of the cells. Yes, it is challenging, but also analyzing those cells comes with distinct challenges. We've done a lot of work over the last 50 years to figure out how best to analyze cells in 2D monolayers. Moving them into 3D poses unique challenges. And so we've spent a lot of time developing solutions, in some cases really detailed protocols around how to do these sorts of interrogations in 3D. Hopefully that means that we can jumpstart you to get to work answering the questions and not optimizing your systems, even figuring out how to ask the questions. So whether that's immunofluorescence or co-culture or large drug screens, we've got some guidance. I'm going to spend the next, 3 minutes or so probably rushing through some of these. The intention is really for this to be a teaser, and I'm hoping that this can be the start of a bit of a conversation here. On the screening side, there is an underappreciated complexity about how to get these cells reproducibly into plates so you can interrogate them and trust the results and not just be looking at noise throughout the system. So the suspension approach has made this very conducive to liquid handling. Nathan talked about how they're doing it, and we're using some of the same tools. Unsurprisingly, you move from purely humans involved to getting some automated liquid handling, and you see your CVs collapse. I'll also use this as an opportunity to call out some specialized plastics. This is not a Thermo Fisher product, but this is a 96-well plate where each of the wells is subdivided into about 100 micro wells. You can see that that does a nice job of controlling the size and the displacement of the cells within the well. You actually now have 100 replicates within a given well. And we think you can get more information out of a single well than if you were doing, say, one spheroid or one organoid per well. So this is a product that we have really fallen in love with. They're not available from Thermo Fisher. There are some of these on Fisher's side, but Corning has some, as does InSphero. So I would highly encourage you, if you're doing these sorts of assays, think about automation and liquid handling, but also think about the plate format and make sure it's aligned to help you answer the questions that you're asking. Really quick shout for the mode of interrogations. Actually, Nate talked about the visual, microscopy-based readouts versus traditional kind of plate reader-based assays, whether they're fluorescence or luminescence. We have done a lot more work lately in image-based readouts, and it's not just visual, like visible light, but you can multiplex fluorescence assays, single well, 100 replicates per well, multiple channels allows you to glean a lot of information from a single well. These cells are precious, and there's a lot of work to get them there, and so we're trying to claw as much information out of those wells as we possibly can. So we still use plate readers. We as a community, I don't think we're ever going to fully move away from plate readers, but we are doing a lot more of high content analysis. And if you're thinking about high content analysis, I would put in a plug for something with confocal capabilities. So we've got a product called the CX7 LZR that allows us to look at these 3D models, not in epifluorescence, but more targeted at individual focal planes. And that will significantly decrease your noise and increase the quality of your data sets. I mentioned co-culture, and I needed to at least get to this slide because it's probably my favorite one here. So what we have done are labeled tumoroids with a red. This is actually not quite a viability indicator. One other practical piece of guidance I can offer: the dyes that we use for 2D cultures are not always perfectly aligned for cells in 3D. In many cases, it's just a matter of changing concentrations or changing incubation times. But in some cases, live cell stains are one example. They often don't work very well over long-term culture. So we've shifted to using mitochondrial dyes as viability indicators. So this is a red mitochondrial dye. The signal goes away as polarity across the mitochondrial membrane goes away, and they're really nice proxies. Here we've got NK cells. Those are those little black dots dancing around. And as they do their work, that red signal goes away, and the green signal, which is an apoptosis indicator, emerges. So there's a lot of work we have done in optimizing the system to enable and allow for this, and have tons of guidance on co-culture models, not just with NK cells. We've done it with T cells. We've worked with fibroblasts. And the last real content I will talk about is a microfluidic device and work we've done with AIM Biotech, looking at vascularized tumor models. So they've got a chip. It's essentially a microfluidic chip it's like multiple microfluidic chips in a multi-well plate format, similar to what we just heard about from Memetis. What this enables is perfusable vascular networks where you can then interrogate some other model inside. So here, this video should be looping. Yeah. So you got a perfusable vascular network with a tumoroid at its core. And if you look at these images on the right, you can actually see that vascular network is invading into, and we hope ultimately vascularizing the tumor model itself. We're in the very early stages of this one, so trying to figure out what exactly questions we want to ask next. But I think it opens the door to some really interesting studies that could be done here. So I'm already a little over time, so I want to thank you for your attention. And before I go, we are going to be offering actually a series of hands-on trainings in our labs, in the R&D labs at the site in Frederick. These are going to be free. If anybody is interested, please drop me a line. You got my email address there. For those that maybe aren't right at the computer and don't see it, it's just matthew.dallas@thermofisher.com. If you or your teams would be interested, please drop me a line. The other plug I will make, tumoroid lines. I mentioned they're hard to come by. PDMR is a really great resource. Look there. We've got a couple large handfuls of tumoroid models that have been built in OncoPro. We've tested them. We know they work. And so if you have interests there, please reach out to me. I'm happy to provide some information. We've got full transcriptomes, full proteomes, and the mutational profile. So if you've got biomarkers of interest, you can overlay what we've done with what you are interested in and see where that may align. All right, with that, I'm going to stop, and I'm happy to take any questions you got. Thank you.
Have you done any work? You've done so many different, you've interrogated these tumoroid cultures in so many different ways. We only have 15 minutes, so you didn't have a lot of time, but have you all done any investigations of like treatment intervals, right? So rather than applying a treatment and leaving it for the entire assay period, maybe mimicking dosing.
So we have not done that, but certainly thought about it and are working with customers who are doing those things. The other thing that we've heard about is wanting to develop models where sensitivity can be, you can manipulate the sensitivity and actually develop resistance and watch resistance to these therapies develop over time. So there's lots of investigations happening there. The challenge for me, honestly, is where do we stop? Because we're developing tools and trying to enable scientists. There are so many amazing questions that we could ask here, and we got to figure out, like, where do we stop and let customers kind of fill in? That probably is one example of something where we have done that. But it's a fascinating question. One other plug I will make for this, I think this is potentially, these models are a platform for personalized medicine. So we have, I think, reason to believe and are developing larger data sets to show that the treatment responses of these cells from these models will reflect how patients respond in the clinic. And that opens doors to changing the way we treat cancer going forward. So the early days here, but I think there's a lot of promise in those sorts of investigations as well.
You did mention Gibco on the first slide.
Yes.
And there's sort of a history with this. Is it still, are there remnants within your company that are sort of the Gibco staff, or is it not integrated?
So it's funny. So I started as a LifeTech employee. We were acquired by Thermo Fisher in 2014. LifeTech, Gibco is one of the key brands of LifeTech. That never dies. So the site in Frederick, our focus is media development. And so that's really all of it under the Gibco brand. So their integration is a continuous process. So we are half Gibco and half Thermo Fisher, but it is alive and well. Yeah, we're still doing a lot there. All right. Thanks, everybody.
Thank you so much for coming, everyone. I would like to thank our sponsors for the breakfast, Burgers and Nipple, and also the Frederick County Chamber of Commerce, because they are our co-organizers of the speaker series. And the next seminar is going to be August 27th, topic to be determined. So please stay tuned and come again, and we'll connect virtually. Thank you so much, Nathan, Paul, and Matt. Those are great talks.